My name is Greenwald. I'm the DaCosta Professor of Biology at Columbia University. I work on notch signaling and cell-cell interactions in C. elegans, a microscopic roundworm. So in my work, I've characterized lots of mutations that affect that process, defining notch genes, notch signaling pathway components, and modulators of signaling. And the general significance of that is that because things are conserved in all animals, what we learn in something simple like C. elegans can be applicable to even human development, and mutations in notch and signaling components can explain certain congenital diseases. But also, aberrant notch signaling is actually a cause of certain cancers. To receive a Canada Gairdner International Award is an overwhelming honor. It feels particularly moving to get this now because of the mission of the foundation to bring science and the appreciation of basic research to people who are not in science. That feels very timely now. So now for my favorite organism, the microscopic nematode worm C. elegans. I think it all goes back to the brilliant pioneer of molecular genetics, Sidney Brenner. He worked out fundamental features of the genetic code using bacterial viruses, and then he wanted to study problems in animal development and neurobiology using similar approaches. So he considered lots of options, including Gary's favorite, Drosophila, and decided to develop C. elegans as a model because it had practical features that he could foresee would enable fabulous genetics. And so, for example, if you put a single hermaphrodite on a petri dish, regular petri dish, with food and come back a week later, you'll see more than 100,000 grandchildren of that single worm on that dish. And so also it hit the sweet spot in between simplicity and complexity in its anatomy and development. So an adult hermaphrodite is a complete little animal with nerves and muscle, no bones. And it has only 959 somatic cells. And that number is precise because the cell lineage is essentially invariant. And because the worm is transparent, wonderful scientist John Sulston was able to sit down at a microscope, watch the zygote divide until the complete organism was formed. It's an incredible feat in itself, and it provides a clear baseline for applying genetics to understanding how cells know what to do. So those are the two features that first attracted me to C. elegans as a student and have been foundational for all the work I've done. The big thing was the space race, and I had fantasies of being the first child astronaut because I had this book called How and Why Wonder Book of Planets and Interplanetary Travel, and I memorized it, and I thought NASA would be so impressed by how much I knew that I could go to the moon. And then when that didn't happen, I got another book called The Human Body, which is where I first learned that we came from a single cell and look at us. And I got interested and read a lot of science books. Another influential book was The Microbe Hunters